I am Michele Dudash, and I am coming to you from South Dakota. So a little bit different geography, and I want to thank the organizers for CNPS, and I also want to acknowledge my collaborators, because I'm going to be telling you about a number of issues and ideas that we've been working on for some time. And I do want to acknowledge that South Dakota State University is located on ancestral territory of the Lakota people. Next slide, please. So this has been, what I'm going to be telling you about, longer than a 10-year collaborative effort. And some of our prior work led us to this textbook, which came out in 2017. So in 2011, we published a review on all the outbreeding and inbreeding depression studies in both plants and animals. And then in 2012, we wrote a paper together about what a species definition we should be using for conservation. And we advocate for the biological species definition or the differential species definition. And then this led us to this textbook. And I'm going to be sharing a lot of ideas that have been developing for quite some time. Next slide, please. So most recently, we published a practical guide for genetic management of fragmented and animal plant populations. It came out a year ago now. And this really does have all the same messages and really practical recommendations about how we might want to think about actively managing our threatened populations and species of plants and animals. Next slide, please. Isn't there? Yes, there we go. Okay, so what we've really been talking about, at least in a number of situations, are the fact that we have had a lot of habitat fragmentation. And this is really the backdrop of a lot of our work. We've had habitat fragmentation. This is a global issue. And we know we need to conserve, preserve habitat so that we have the opportunity to try to manage. And we also know that some populations always have been small. So with smaller populations in this habitat fragmentation, independent of how you've gotten there or how long you've been there, today we're probably looking at inhospitable matrices surrounding these fragmented populations. And we know that we've played a large role in this. Next slide, please. So there are genetic consequences to fragmented populations and distributions of species. First of all, there's often reduced or no gene flow among the fragments. And we know from our work that over 55% of plant species are showing evidence of inadequate gene flow among populations. So when you have a lack of gene flow, you tend to have more mating among relatives because you're a smaller population. With that comes inbreeding and then inbreeding depression in many instances. And we have pretty much demonstrated, along with many others in the last 10 years, that inbreeding depression is pretty ubiquitous in nature. With inbreeding comes this loss of genetic diversity. We see a reduction in reproductive fitness or traits that would be correlated to reproductive fitness. We know that this does elevate extinction risks, and it also decreases the population or the species' ability to evolve, or whatever taxonomic unit you're working with. Next slide, please. So what do we recommend? Well, we actually recommend, next slide. We recommend active management of populations. So we are advocating for reestablishing gene flow or translocations, or the other term is genetic rescue. And I think we've heard them all earlier in the day. So if we establish genetic rescue and actively manage, we are going to reduce inbreeding and the resultant inbreeding depression in many instances. We're going to increase or restore genetic diversity of the population or the species, the unit in question that we're trying to manage. We're going to improve reproductive fitness and overall population demographics. We're going to reduce the extinction risk, and we're going to increase the ability to evolve through with climate change that we know is ongoing. And we heard about some pretty interesting studies this morning. But to date, there have really been very few intentional genetic rescue studies conducted. Next slide, please. So how do you measure genetic rescue? So we have heard this morning of many studies this morning that had really detailed genetic data. Well, you can measure genetic rescue by using any trait that you can measure, and it doesn't have to be a genetic marker. So you're basically looking for the % change in fitness between your outcross and your inbred progeny. So this differential performance between outcrossing and inbred progeny would allow you to understand the magnitude of how you are improving, if you are, by augmenting gene flow from another population. Next slide, please. So there has been, this is by Dick Frankham, and Dick Frankham led the 2 books that I have been involved with, and he's also written a great book in conservation biology with John Ballou and others. But he did a meta-analysis on all the genetic rescue studies that he could use to estimate genetic rescue of plants and animals. And he did 156 comparisons. And I've had the pleasure of working with Mimulus in Northern California for many years. And I've also had a chance to work on a self-incompatible daisy. So 2 of those yellow, beautiful things in there. And he did 156 comparisons he looked at. Next slide, please. And what did he find? Well, he found that outcrossing of inbred populations resulted in large beneficial effects. So the genetic rescue was over 90%. In over 90% of the cases, he saw genetic rescue effects positive. The median increase in fitness, if we think about fecundity and survival, is 150%. And the genetic rescue per generation is over 20%, and it had a broad range. And he also found that the impact of genetic rescue persists across generations. Next slide, please. So what are we advocating for? We really want to have you think about active management. So we can either watch taxa go extinct owing to the fear of outbreeding depression, or we can do something about it. At least consider doing something about it. And when we're talking about that, we're talking about gene flow augmentation, translocation, pollen seed, whatever you want to do. And we advocate for this because we think we have pretty strong grounds and data to show you that outbreeding depression is predictable in many instances. Next slide, please. So here is our decision tree, and this was published in 2011, and it's also in both of the books. So here you can predict outbreeding depression. So let's walk through this. First of all, is the taxonomy resolved? So do we know what we're dealing with? And again, we advocate for the biological species concept, or at least have folks tell you what species concept they're using. I mean, there's probably close to 30 of them now. There were 26 a long time ago. Fixed chromosomal differences. We know in that daisy that I worked on, we have diploids and polyploids together, and you can't tell with your naked eye. So are we dealing with the same thing? And then is gene flow. So we go to number 3, gene flow between populations. Has it occurred in the last 500 years when the human population has increased? And if the answer is yes, we've had gene flow, then you go down to significant environmental differences. Are there significant environmental differences? And this is where I think we have the biggest point of discussion, because we think of environmental differences more like growth zones for our vegetables and our tree plantings, for our gardens, and maybe we should be thinking a little more broadly. Obviously, we heard about how unique California is with their serpentine soils, and there are instances where you're going to have local adaptation that might prohibit this, but we do allow for that. So if there are significant environmental differences, we say you should, then you ask, have the populations been separated by greater than 20 generations? That's the next question you asked. And then if they have been, you might be able to consider it. But this decision tree gives you an idea of whether you should predict, whether you're going to predict seeing outbreeding depression. So you have a low risk if you've had all of those differences which are not happening in terms of gene flow and environmental conditions, then you have a low risk of outbreeding depression on the right-hand side. So there is a way to get a handle on this, to think about whether you should consider it. Next slide, please. So the 3 things I want to highlight for outbreeding depression that keep appearing in the literature are the major chromosomal differences. So you have the diploid and the polyploid populations within the same general area. And this could be a challenge, long isolation, greater than 500 years, and then the adaptive differentiation. So this local adaptation and this non-adapted uniqueness. And we do know that even when you've done long distance crosses between plant populations, even if you do see outbreeding depression, following additional generations of crossing in natural populations, it goes away, it's mitigated. So if we're advocating that if you give them genetic diversity, things are going to happen across generations. So that we think that's really a critical thing to consider. Next slide, please. So here is what we're advocating for is a science-based management of fragmented populations. So here you can think about if there are multiple populations, if there is only one, are there any other populations you can cross to? Will crossing lead to outbreeding depression? You can ask this. Will crossing result in worthwhile genetic rescue? We've given you ideas of how you can measure this and quantify it so you can make knowledgeable decisions. And we're always considering whether the benefits outweigh the costs. Next slide, please.
1 minute.
Thank you. So is gene flow going to result in worthwhile genetic rescue? It probably will in many instances. So so in terms of if there's a lot of inbreeding depression, if the recipient or the donor population is unique, and if the benefits outweigh the costs. Next slide, please. Is gene pool mixing a good idea? Well, how best to do this? Well, you really do have to think about donor population. So I would say if you can have more than one donor population, that is better. You watch the number of individuals. When do you stop? Yes, we're concerned about swamping, but these are things you can think about, but you have to know the biology and the ecology of the species in question. Next slide. With minimal information, I just gave you that, so you can skip that slide. And with the luxury of detailed information, which we have seen this morning, you can ask more specific questions. But either way, populations should be monitored to confirm that augmentation has resulted in intended benefits, and you want to monitor the demographic responses of populations to augmentation to see the impact of what you have done to the population. Because remember, the goal of all of this work in many instances is to have self-sustaining populations where you don't have to go in and augment in the future. Next slide. So we're really advocating for a proactive management, same karyotype, similar environments, gene flow with the last 500 years, at least consider augmenting so we don't lose biodiversity. Next slide. Here's my take-home message. So we do think you need integrated management approaches. Take as much information as you have to make your plan. We really think doing nothing is a choice that is harmful because species are not static, they're evolving. And we have to, if we provide the genetic diversity, we might not have the exact same species, but if we have something that serves a community or ecosystem, then I think we've succeeded. And next slide. Thank you very much.
Awesome, thanks, Michele. So we have a few minutes for questions, and attendees, you can use the chat function to type in your question so that we have a record of it. Or you can raise your hand and we'll maybe pass the mic to you as well. So Michele, looks like there's a question about how can we be sure we are rescuing versus contaminating?
Okay, so I think you need to know what you're working with. If you think it is the same species or whatever you want to call it, species, subspecies, and you have low genetic diversity, and you see the harmful effects of inbreeding, I would say that it's better to try than not try. So I can't, without specific information, I don't think we can tell, I can tell you that, but I do think it's important to try to watch things go extinct, even though we know things go extinct regularly. I think if we try, we might be generating a new population. So, or maintaining a population that plays an important role. So it's very hard to know. But the risk of outbreeding depression, there really isn't a lot of evidence for outbreeding depression for any long-term effects. And there are a number of studies that have demonstrated that.
Awesome, thank you. We'll go ahead and continue on to Amy, and we'll still have time at the very end for a panel discussion. So if anyone has more questions for Michele, you can ask at that time. So welcome, Amy. Go ahead and introduce yourself.
Thank you.
Thank you. Thanks, Michele. It's a great talk. And I am going to continue on with what will hopefully be maybe a practical example of some of these questions in practice. I'm Amy Vandergast. I lead a population genetics laboratory in San Diego for the Western Ecological Research Center of the USGS. And I've been doing that for about 15 years now. Prior to that, I got my PhD at Berkeley, but in the insect science department. And I've worked on a variety of questions in taxa, primarily on endangered and rare species, but mostly animals. So plants are a little bit new to me. More recently, my lab's expanded to include work on plants, and this study on salt marsh bird's beak we initiated at the request of San Diego Association of Governments. They have an MSCP plan, and this is one of their listed species, and then the Navy, who also have populations on their property. So they're interested in learning more about the genetic structure of the populations on their land and how they relate to the rest of the species or subspecies range. So luckily I get to work with more experienced botanists. Sorry, you can stay. I'm not really ready to move on yet. I wanted to introduce my co-authors, Dr. Liz Milano. She's a plant geneticist. She's been a part of my lab group at USGS for the past 4 years. And then Margie Mulligan and John Redman are also an integral part of this project, and they conducted all the field work and collected all the specimens. Okay, now we can move on to the next slide. Next slide. Great, okay. So sorry, go back one. So I know we've said this multiple times already in the conference. Genetic diversity provides the raw materials for adaptation and evolution. And in outcrossing species, fitness is positively correlated with genetic diversity. So for annual plants, I think there's importance in understanding both what that standing diversity is and the above ground plants, and then even more so in the seed bank. So we do need to understand what are the natural extent of both pollen and seed dispersal among discrete occurrences. And we should characterize this in order to understand whether this natural connectivity has been disrupted by human landscape change and thus would require management. And I think the flip side of that, you know, at the same time, we would want to avoid swamping locally adapted genotypes and diversity with overmixing strategies. So we're trying to understand a little bit about the history of these populations and interpret the data with that in mind, as well as what's going on currently. Next slide, please. So we can characterize sets of populations in terms of some simple measures of genetic differentiation among populations, diversity, such as heterozygosity within populations, and then understanding inbreeding or maybe relatedness within populations as well, and use these data to provide recommendations to management. And I threw up a figure here from Ottewell and others. This is from a 2016 diversity and distributions paper that they put out, which gives a nice general flow chart on how to relate these genetic measurements back to management strategies. So for example, in a system where we would have low genetic differentiation among populations, high diversity and low inbreeding, we might infer that this is a really intact system and it would need less intensive genetic management or restoration. Our next slide. And the converse of that would be these small, isolated, and homogeneous gene pools, which could require more intensive site management, possibly, as a first step to try and recover diversity in situ, and then more careful research may be needed if seeds need to be brought in from another population source. Next slide. Okay, so this is our study system. Our taxon of interest is the salt marsh specialist, Chloropyron meridimum meridimum. Its distribution is currently restricted to just a few salt marsh systems from Morro Bay in the north, south, into Baja California. It was listed in 1978. We know some things about the species, but maybe not as much as we need to know. It is an outcrossing annual. It's pollinated by solitary bees. Seeds can float, and so dispersal is thought to be facilitated by tides. It is a hemiparasite site, but it has been found to use an array of different host plants. Population sizes undergo annual booms and busts. They can range anywhere from dozens of plants to thousands of plants. And some long-term monitoring work in San Diego has associated these boom and bust cycles with tidal amplitude cycles and then spring rainfall and temperature regimes. So as a salt marsh specialist, we have to recognize that this subspecies is at very high risk of habitat loss due to sea level rise, particularly in marshes where there is no place for the marsh to retreat. So because of this, restoration, augmentation, and reestablishment will likely be really important management strategies for this subspecies in the future. Next slide. So this is a map of both the subspecies range and where we collected genetic samples from. So we collected from up in the north, Morro Bay. There's about 3 occurrences known up there, a couple of occurrences at Carpinteria. There is a group of more occurrences in Point Mugu and Ormond Beach. We were able to sample those, as well as in Newport Beach and down in San Diego Bay. Mostly the natural sites there are in the Tijuana estuary. There's a site at Dog Beach and the Sweetwater Marsh are both restoration sites. And then we were able to get samples as well from the Baja populations down in Bahia Falsa and Punta Azufre. So that was nice. So there were some sites that were known in the LA basin and in northern San Diego and southern Orange County that are now extirpated or at least haven't been seen for a long period of time, as well as some inland sites. But those are also no longer extant as far as we know. Next slide. So really briefly, our methods here, we collected from each of these sites, including some information on some of the field covariates, such as soils and co-occurring plants, population size estimates, and some observations of different growth forms. Our genetic method that we used was a double digest RAD-seq protocol to sample genes from throughout the genome. And then we also used some flow cytometry on a subset of 5 samples per site to assess genome size differences. Mostly we were interested in determining if there were major differences in ploidy, either within or among sites. Next slide. So I'm gonna fly through some results for you as well here. So this is a structure plot. Each bar represents an individual in the analysis, and the colors represent the assignment to a genetic cluster. And we found really strong genetic clustering among these 5 regional bay groups. We had Morro Bay in the north, We had Point Mugu and the Carpinteria sites clustering together. Newport Bay clustered out separately, the San Diego sites clustered separately, and then Baja as well. And, you know, applying hierarchical F statistics in an AMOVA, the large majority, 73% of the variation was partitioned among these clusters, and a much smaller amount, only about 6%, was found among occurrences within clusters. Next slide. So just showing this in a slightly different way. This is a maximum likelihood analysis of concatenated sequence data and individuals grouped together according to their regional groupings. And I also wanted to show some of the different growth forms and variation that Margie Mulligan noted when she was collecting in the field. So she noted that there were some variation between sites in both the branching characteristics and the bracts. These are some of the characteristics that are used to distinguish subspecies in this group. And so one of our recommendations is following up with a more detailed morphological work to try and quantify this across the subspecies range, and then also with the other subspecies within the group. Next slide. So I'm not going to spend too much time here, just to say that genetic diversity, you know, varied a little bit between occurrences, but generally was higher in the southern portion of the range in those Tijuana estuary and Baja California samples. We didn't see any evidence of elevated inbreeding within any occurrence. The coefficients were close to 0, but that being said, our error bars were really high. So maybe we still need to do a little more work to figure that out. Next slide. So this is just another way to show you that there was a latitudinal trend in diversity and also in genome size, which was interesting to us. Both diversity and genome size were correlated with latitude, and both were higher in the south, and genome size was more variable in the south. So we had some individuals with higher and lower genome size there than elsewhere. So we didn't find any doubling of the 2C values that would indicate different ploidies. So rather we think that the difference could be due to supernumerary chromosomes, and those have been known to contribute to diversity, although again, this would need to be followed up with some microscopy or other studies to see what's going on there, and potentially looking at more individuals per site as well. Next slide. So even though we're just really brushing the surface so far on the genetic structure and the significance of that across the range, we still can use our information to provide you know, some guidance for conservation strategies moving forward, and then also to identify potentially compatible sites for seed sourcing if needed in the future for restoration. The U.S. Fish and Wildlife Service recovery recommendations include protecting occurrences within major marshes within the historic range. And our results confirm that this is an important strategy, and that those protected marshes should include, you know, some within these different 5 distinct genetic clusters, because each of these regions contains distinctive genetic variation that should be protected. Next slide. So maybe we get into trickier territory here. For seed sourcing with the data we have in hand, we could consider 2 basic genetic factors: the genetic similarity among sites and the genetic diversity. Because we found relatively high genetic similarity among most occurrences within clusters, you know, it seems like a reasonable approach to continue restoration efforts that are close to existing extant populations, sourcing seeds from within those same regions, and then targeting the largest or multiple local sites to maximize the diversity there. And that approach has worked reasonably well. Sweetwater Marsh is a great example of a restoration site, a population that's you know, been restored and it has been there for 20 or more years on the landscape. So I consider that a success. You know, however, it could be trickier when there are reestablished efforts that are happening between extant clusters, since we do have, you know, large spatial gaps and potentially large genetic differences among these clusters. We have the Newport cluster, the points in red there in the middle, and the star there is a Huntington Beach or Bolsa Chica Wetlands Restoration Project, and it's currently sourcing seed from Newport. That site is not doing as well, although it's only in the first couple years of attempts. But it could be reasonable to test out mixes from some of these different regions, and particularly focusing on those that have traits that could be useful for future persistence. And I would say for this plant, that could be greater temperature salinity tolerance. So we don't really know if that's the case, but I think that's data that we should try to gather from around the range to see if there are differences and if those correlate with any genetic differences. Although given the amount of differentiation that we did measure, I think I would also recommend that these approaches be tested pretty stringently before applying in the field. So we'd recommend test crosses to assess compatibility and fitness before any mixing. And finally, I just want to end and say that, you know, genetic considerations are just one of several in identifying these compatible seed donor and receiver sites. It sounds like Andrea will kind of continue on this theme in her talk as well and explore this topic some more. But habitat conditions, host plants, morphological similarities might also be important to consider for salt marsh bird speak. And final slide. So I'm probably running over time. I tried to fit a lot into this talk. So there's a lot more I could say, but I'll wrap up just with these acknowledgements. Thank you.
All right, thanks so much, Amy. We are going into our break time, so we'll go ahead and skip verbal questions, but feel free to use the chat function to ask any questions, and then we'll have that closing panel to ask then as well. So we'll be on a break until 2:10, just a little under 7 minutes at this.
I'm Andrea Kramer at Chicago Botanic Garden, speaking today about considerations when mixing source populations to restore rare plants. Just want to acknowledge my collaborators on this project. Adrian Basie St. Clair was my graduate student who led a lot of this work. Peter Dunwitty at University of Washington and Tom Kay at Institute for Applied Ecology did all of the background work on these species that allowed us to conduct this study, and then Jeremy Fant also at Chicago Botanic Garden. So let's jump right in. Next slide. Why mix source populations? A lot of this has been covered, so I'll go over it quickly. You may want to mix source populations if you don't have a source that matches your reintroduction site, either climatically or ecologically, and you want to hedge your bets. You don't know what may work, and you want to provide raw material for selection to act. You may also be dealing with smaller fragmented source populations, so they may have low genetic diversity, inbreeding depression, or they may not even be producing seed because they're self-incompatible and there are no potential mates there. So clearly that's a must mix situation. And as Michele so nicely pointed out, outbreeding depression is often predictable. So choosing source populations should certainly be taken with a lot of information on your study species, but can be done in a way that is predictable. Next slide, please. So what I'm talking to you about today is thinking about mixing source populations and looking at a study species, Castella Levasecta, that provides a great example of what kind of information you may want to have in hand before you decide to mix, and then thinking about how you actually mix source populations for a reintroduction. And how those different approaches may influence the outcomes of your reintroduction. So Castella Levasecta is a fantastic study species, not only because it's beautiful, it's native to grasslands in the Pacific Northwest, and so there are not many of them left. They are highly fragmented. The picture you see here is one of the remaining populations on a very steep slope on one of the islands in Puget Sound. There are only 10 populations left on the islands of Puget Sound and one remaining population on the mainland of Washington State. So it's been extirpated from Oregon. It's federally threatened, and the recovery plan calls for reintroduction throughout its range. So this species kind of ticks a lot of boxes. Much of the reintroduction that will need to happen has no source populations that are nearby. And work that has gone on prior to my involvement in this project by my collaborators has shown that there is low genetic diversity in the remaining populations. The individuals in those populations are experiencing inbreeding depression. Controlled crosses show that the offspring are more fit than either of their parents from individual populations. And common garden studies that have been conducted in the Willamette Valley, where the species used to occur in Oregon, have shown that not all potential source populations perform similarly at those sites. And the most likely, the best matches are those that are most similar ecologically. So this provided a great, it was very clear that in this case, reintroductions really required mixing source populations, and all of that great background work, next slide please, allowed us to conduct the study that I'm talking about here. So starting in 2008, after all of this background work was done, the decision was made to mix 4 wild source populations to provide material for reintroductions. And because the reintroductions needed to happen on a large scale with a lot of seed, an additional step of mixing the material in a nursery setting to get sufficient outcross seed was deemed necessary. So those 4 wild source populations were chosen because they either performed well in those common garden sites in the Willamette Valley. That was the populations N, E, and C, both came from island populations. And the R population came from the remaining mainland population in Washington state. It actually didn't perform particularly well at the Willamette Valley site, but their decision was made that they didn't want to lose those genes since all of the reintroductions were occurring on mainland sites. But particularly in Oregon, they wanted to minimize the potential representation of that population. So they planted fewer individuals from that R population than the other 3 when they planted the nurseries. So this shows that there are, in Washington, the decision was made to mix the 4 populations together and plant a nursery without keeping the populations separate. And then the reintroductions were conducted following I have a cat in the room, sorry about that. The reintroductions were conducted using seed from that nursery. In Oregon, the decision was made to keep the individual source populations separate in source identified rows, but allowing outcrossing to happen between the rows. And then mixing happened following seed production in the nursery. And plugs were used to conduct reintroductions in Oregon, as well as seeds. So this provided a really great way for us to look at how genetic diversity and inbreeding and individual source populations flowed through the production and use of mixed materials for reintroduction. So just to help orient you, each of these boxes represents a different population that we were assessing, and we used neutral molecular markers. We used microsatellite markers to track diversity through this process. So if you go to the next slide, we identified kind of different clusters that we wanted to pay particular attention to through the process. The red were the wild source populations. Nursery populations are outlined in green, and then the 3 different types of reintroductions using Oregon material that were plugs, Oregon material that was seed, and then Washington seed. You can see each of those different colors. The questions we asked were whether diversity and inbreeding really did change through this process. And all source population, we wanted to determine whether or not source populations were represented through the process and how. And then also whether the production approach that was used, either mixing before the nursery or after the nursery, influenced how diversity made it through the process. And then finally, whether or not the propagule type influenced all of those measures. So next slide, please. Okay, so you'll see the schematic up in the top there. With our molecular markers, we were able to determine that yes, genetic diversity did change through this process. It was greater in reintroductions in the wild or the nursery population. So you can see the red and the green boxes had lower diversity. In this case, this was observed heterozygosity, and it was much lower than the heterozygosity, the diversity in any of the reintroductions. And thinking about or looking at whether production approach when you mix or the propagal type you use really influenced the level of diversity that came through. Maybe is our answer. We did note that the highest and most consistently high diversity was found in the reintroductions that used seed from the Oregon nursery where mixing happened following production in the source identified rows. Next slide, please. So we can use these data to also look at inbreeding to determine whether or not it changed. We had hoped and predicted that it would, and it did indeed change through the process as expected. Both on the slide here, we've got inbreeding, which is F, as well as relatedness, kind of two sides of the same coin, showing that our wild populations had not terrible inbreeding, but we know they were experiencing inbreeding depression. That's a separate point, but they were not particularly inbred, and they were intermediately related to each other. In the nursery, we actually saw increased inbreeding and decreased relatedness, likely because the conditions in the nursery were not stressful. And so some of the more inbred individuals in our seed lot from the wild populations were able to survive than they would have in the wild. When we get to the reintroductions, you can see that the level of inbreeding decreases as predicted, really throughout our reintroductions, and relatedness also decreases. And so again, it looks like that blue box, the seed produced in the Oregon nursery, or the seed produced in the Oregon nursery and used in the reintroduction as seed, really delivers kind of the most consistent levels of low inbreeding and low relatedness, as well as high diversity. Next slide, please.
1 minute.
And then finally, we were able to track the wild source population. So this is similar to the slide that Amy showed. Each box represents our populations, each line is an individual, and each color represents a genetic cluster that was identified. So you can see conveniently for us, our wild source populations have different colors. They grouped differently from each other. And you can see in our Oregon nursery, they were fairly consistently represented. Our sampling occurred 3 years after the nursery was planted, so we see that there may be some self-sowing happening there. But for the most part, we're seeing separated individuals. And then when you look at the reintroductions, you see all of the colors represented, all of the source populations represented. And importantly, the R population, that one that was from the mainland but didn't perform particularly well in Oregon, was there, but not in a really high abundance. When you get to the Washington nursery, where the mixing happened prior to the nursery planting or in the nursery, you see that we actually largely lost representation from one of the source populations, the N population, that kind of light yellow. And that kind of carried through. The seed reintroductions were much more unpredictable. Some showed much greater of the blue, dark blue, R population, and some did not at all. So a lot of inconsistency. And again, we find that those Oregon seed reintroductions may be kind of the best performers. I will note that there's a little more variation in which source populations are represented there. And we think that may be the result of selection that's occurring on the ground. You put the seed out and you let the site kind of filter which individuals are most likely to survive. So just to tie this all back together to creating reintroductions that can persist, the work of my collaborators have shown that the only consistently demographically viable populations that have been performed using seed. That's both in Oregon and Washington state. So kind of bringing it all back. All right, last slide, please. So the conclusions are that mixing has worked. The aims have been achieved. The timing that you mix really matters for being able to control what makes it into your reintroduction, and propagule type may matter, with seeded reintroductions really being the most likely to produce demographically viable populations. Next slide, please. All right, and this is just a thank you to other folks who helped us get this going. And then if you go to the last slide, there's the citation for the paper that was recently published in Restoration Ecology, as well as other citations that I thought might be useful on this subject. So now I can take questions, or I'm sure I think I'm over time too.
Great, yeah, we have just 1 minute, so if anyone has questions, it looks like we just got one from Naomi, possibly. It says, or she was talking to Andrew. Are you able to open the chat, Andrea, to see that, or do you like me to read it?
Target restoration. Well, the target restoration population, I guess I'm not quite sure. The source populations were included in the study. Oops, all the source populations were included in the study. They did have lower diversity and higher inbreeding than was desirable. I'm not sure if I answered your question. If I answered your question, Naomi.
All right, great. I think it cut out a little bit there, so we'll maybe have you answer a little bit more later, but we'll move on to Loraine. Thank you, Andrea. Great. All right, go ahead, Loraine, and introduce yourself.
Hi, thanks. Thanks, Anne-Marie. First of all, I want to thank CNPS for bringing this really interesting meeting together and for including me. Myself, I'm a botanist, conservation biologist who's worked in a lot of different capacities in research, teaching, and conservation. And I'm going to be talking today about work that I did while I was at the California Botanic Garden in Claremont, where I managed the labs and did genetics research from 2013 until last year. I'm now zooming in from Maine. Next slide, please. So what I want to talk about is the role of time and the uncertainties that we face while examining genetics in rare plants. And in the context of applying genetics tools to translocations, we're really very intentional in our efforts to keep every cog and wheel in our intelligent tinkering, using our best available data. Yet we do this in the present moment, caught between our shadowy understanding of the past and an equally foggy sense of what the future brings. So by the shadowy past, I'm thinking of changes in Southern California landscape since the Pleistocene, and of changes that native Californians brought to plant distributions and abundances, and then of the many rapid changes to the land over the past 170 years. This past has shaped the plant populations that we see today. And we then make a snapshot genetic assessment using the magic of lab work. And in conservation, we then take action. And translocations represent a gamble on an uncertain future infused with hope, but where there are many potential changes coming down the pike for us, too many really to consider them realistically. Next slide. To illustrate our condition as humble researchers in this grand temporal epic, I'll be pointing to 3 taxa to exemplify translocations that happened in the past, which are happening more or less in the present, and which will happen in the future. So we'll be looking at Berberis navinii, at Hazardia orcudii, at Berberis pinnata subspecies insularis. These are all rare plants. 2 of them are listed as endangered, and they're all CRPR1b species. Next slide. So as the first example, Nevins Barbary, it tells a tale of how a chance translocation of the past secured the future of a Southern California endemic shrub, the Nevins Barbary. And there are at present just 2 somewhat substantial populations of this species, and one of them resulted from a translocation nearly a century ago. Nevins Barbary was first collected by Nevin in 1882 in the San Fernando Valley. And even by that time, the plants that lived in the San Fernando Valley had seen plenty of changes. So we can ask, what was the backstory of why Pacoima Wash in the San Fernando Valley was one of the final holdout locations for this rare species? Well, we don't know why, but we know that there is a story. Next slide, please. So there are most likely fewer than 500 individuals extant in the wild. And it's also not an uncommon horticultural species admired for its flowering displays. And the odd thing is that it can and does escape from cultivation to some extent, which begs the question of why it's not more abundant naturally in Southern California. The genetic study that I did used microsatellites to examine all natural and some escaped or possibly planted plants. And I'm not going to discuss the results in detail today, but just give some broad conclusions from them. Next slide. This map shows the CNDDB localities for Berberis Nevinii. And the main thing I'd like you to notice here is that there are two somewhat substantial populations of this species. One is at Vale Lake in the south by Temecula, and the other is in the north in San Francisco Canyon by Santa Clarita. The type locality from the San Fernando Valley was a population that was extirpated in the late 1930s. So one question we might ask is, what is it from the shadows of the past history that accounts for this distribution? And why is it so spotty? We don't know the answer to this. And the big headline from this slide, though, is about the translocation of numerous plants from the San Fernando Valley population, which was subsequently extirpated, from there to San Francisco Canyon, some 30 kilometers north. Next slide. Well, no, actually, the main headline here is that is about a disaster. It's from 1928 in the St. Francis Dam disaster. This was a tragic event early in the history of bringing water to Los Angeles. A dam collapse that happened in the middle of the night and killed more than 400 people. But the odd botanical plot twist of this tragedy was how it resulted in an unintended rescue of the San Fernando population of Berberis Nevinii by translocation, though it wasn't called this at the time. There's a well-known Los Angeles plantsman, Theodore Payne, who was asked a few years later to revegetate the scoured site below the dam. And we know from a letter that he wrote that he had used plants he had propagated from the San Fernando Valley, and even some that he had dug up himself from the original site. So then decades passed following that event, population recruitment happened, and the species was federally listed as endangered in 1998, with San Francisco Canyon given as one of its populations. But ultimately, the translocation with San Francisco Canyon given as one, translocation was a consequence of Payne's just being fond of this species and happening to secure the planting contract. So genetically, the population in San Francisco Canyon is descended from the San Fernando population. And also note that the genetic diversity in San Francisco Canyon encompasses the full extent of known genetic diversity in the species. There's some level of distinction between these plants and those of Vale Lake, but there's really a lot of overlap. Next slide, please. So then there was a fire in San Francisco Canyon, in particular the copper fire of 2002. So we know that it did kill off a bunch of Berberis seedlings, but also as part of this story, the resulting litigation was able to provide funding for studies and restoration efforts. So from demographic work that Naomi Fraga and I conducted, if you see in the map at the left, the plants that we found, we know that the population is increasing, and perhaps slowly, and it's not spreading much away from the original planting site, but it has persisted from the time of Payne. So this is an unusual case where we can see results from a long experiment in translocation. The funding then also presented opportunities for restoration. And we could use these genetic results to inform the San Francisco Canyon restoration plan, with the aim to maximize genetic diversity of the outplants. So we collected seed from the site. We kept track of the maternal lineages in propagation efforts. Going forward, the outplanting will be planned and intentional, and it's due to start anytime. And then going forward from there, the monitoring will keep track of maternal sources. In the map in the lower right, the blue dots are the proposed restoration sites. Next slide, please. So what are some of the questions and lessons that we get from Berberis nevinii? We can ask whether the current distribution is a reflection of past climatic conditions and whether this species was moved or managed by people prehistorically. It was just a quirk of history in the past that led to the translocation to San Francisco Canyon in 1932. So this was like a rescue at the time. And then it's a quirk of history 70 years later that permitted restoration and further translocation to the San Francisco Canyon, and maybe this is rescue number 2. The future of the plants at Vail Lake, which you're seeing in the pictures on the right, is less certain and requires some vigilance from US Fish and Wildlife Service. But that's another story. And but what we have found is that the genetic data has been a useful tool for the conservation of Berberis nevinii. Next slide, please. So then the second case is more typical of translocation, and I'll be giving an extremely abbreviated account here. So the case of Orcutt's hazardia orcuttii, which is a coastal sage scrub endemic, mostly found in northern Baja California, but there is a single population in San Diego County and Encinitas. And I used ISSRs in this study to compare the genetic composition of 4 outplanted populations with the single native US population. And the outplanting here had been done about 10 or 12 years prior to the genetic analysis. Next slide. Next slide. Okay. So this gives the landscape context of this native population. You can see that it's in a very small natural area surrounded by development. And the population itself lies on about 1.5 acres and includes some 670 individuals. In the map on the right, you see the native population in orange and the 4 outplant sites in yellow here. And basically the genetic analysis showed that the 4 outplanted populations just differed genetically from the native population, but the conservation implications of that were actually not readily assessed. So whether that was a function of the propagation process or of any selection that happened post-planting, for example, it's not very clear. Next slide. So, but the 2 main findings that we offered were that it's important to sample seeds from a diversity of individuals from the native population for restoration propagation. And our finding was consistent with a seed collection from a limited number of individuals initially. And it's also important to keep track of the maternal lineages during propagation efforts and planting out to try to maximize the diversity in a situation like this of outplanted populations. So you don't know what genotypes may be best suited to the outplant site, so it's better here to hedge your bets. And the more interesting story here turned out to be the real possibility of longer-term extirpation of the outplanted populations through introgression. As orcuttii, where it comes into sympatry with Hazardia squarosa, Grandilioides, forms hybrids. And so this was a more important message, we thought, to the site managers. Next slide. So then thirdly, I want to look at a translocation yet to be. And here the genetics were used as a step, and that is one of many steps on the path to planning towards eventual translocation or augmentation in the future. And the stories of a very rare island endemic subspecies, which at the start of this study had 5 known individuals on the island and 1 ex situ on the mainland. And this is part of an ongoing project to restore the island Barbary to Santa Cruz Island and perhaps also to Anacapa and Santa Rosa Islands. The initial objectives were to determine whether there were any other genetic individuals of this taxon which might be located in botanic gardens, and also to answer whether about questions about clonality. So whether what looks like a clump or a clump of stems in the field might be more than one genetic individual. And I used microsatellites to examine this. Next slide. So the basic findings of this study showed, I think, the value of being able to distinguish genetic individuals when especially when there are so few of them remaining. First, we found there were no additional genetic individuals in gardens. And in fact, we found that in gardens, there were only 2 individuals that had ever been sampled on the island. And one of those was that clone which is now extirpated on the island, so only exists on the mainland. Also, after some more individuals were discovered on Santa Cruz Island after this, after that initial study. And so genetic analyses there determined that among the numerous stems that had been located, that this added just 4 genetic individuals to the tally, leading to a present tally of 10 genetic individuals for this taxon, including the one that exists ex situ. In the lower right-hand map there, those yellow circles enclose genets of the species of the taxon. So next slide, please. So the island Barbari is just one species in a whole cast of characters in Channel Islands restoration, as I'm sure you're all aware. This project involves many partners and a lot of effort. So it may be a long shot, and there remain a lot of logistical hurdles to cross before the working group is ready to introduce more plants to the islands. But the goals are basically to propagate, to make cuttings of all the island genotypes, to propagate them at Santa Barbara and California Botanic Gardens, and then to cross them, because you probably can't appreciate from the map, but actually breeding is problematic on the island. Most of the individuals are too far from the other individuals for any crossing to happen. And then ultimately to repatriate to the islands to those offspring or clones. That's the ultimate goal. Next slide, please. So in wrapping up, what lies ahead for the use of molecular genetics data in making management decisions for rare plants? So clearly from today's meeting, we can see that the use of these methods are really very helpful, and depending on the situation. We always have to ask whether these methods, we always have to ask whether this is the most important use of limited funds. And we always have to ask whether these data will answer a question that needs answering for the conservation of the species. So we don't want our expectations to be too great merely because the tool is a high-tech one, when the situation may warrant something that's more muddy boots and pragmatic, like fencing or invasive species control. In the hard times coming down the pike, in terms of changing climate, altered fire cycles, impending habitat degradation, the uncertainty of the future is the elephant in the parlor, but one which is really problematic to acknowledge. What might be the consequences for performing translocations and assisted migrations informed by genetic studies? We don't exactly know, but we know that we need to act now and that we need to be as best informed as possible and then to hope for the best. Next slide, please. Next slide. Oh. So thanks to everyone. Thanks to my colleagues at California Botanic Garden and at USGS, and also many, many field helpers and lab helpers that contributed to this. Thank you.
Awesome, thank you, Loraine. So if we have any direct questions for Loraine, we can open those up, but otherwise we'll go straight into our time with all the panelists. So if you were a panelist speaking, go ahead and turn on your video. and unmute your microphone as well. So if there's any audience questions, you can write them in the chat, or you can raise your hand and we'll call on you to unmute yourself as well.
I'm not able to turn on my video because the host has stopped it.
Okay, I'll fix that for you. 1 second.
Thank you.
Okay, you should get a prompt to start it.
Yep.
There we go. Sure, everyone's crafting their perfect questions right now. But you know, feel free to kind of bring up a subject you like. If panelists, you have a topic you want to ask each other as well. We have about 15 minutes before we'll break.
I think it was a really nice balance of talks to really highlight some of the issues we need to be considering if we're going to do active management and how thoughtful a process is. And I really appreciate it, the clarity you brought to it, and when perhaps it's appropriate and when it isn't, since I have no idea what I said. But I found it really helpful to hear what everyone's doing, and it's very exciting work and challenging.
And Loraine brought up working with Naomi, who I believe is on our final panel this afternoon.
Brent, you can go ahead. Thank you for raising your hand, both virtually and in real life. Go ahead.
I wasn't sure which one was the way to go, the little blue hand or not. I just wanted to be sure that we circle back to these issues, the differences between plants and animals, and a lot of the conservation genetics research is done on animals, and we're trying to import it into plants. But as I was commenting along in the chat on the way, plant species are quite a different thing than animal species. They're certainly not biological species. If there's one concept that does not fit plants, it's that one. We have much more local differentiation than would be typical in at least a highly mobile animal species. So none of the named species in plants, basically, unless there's only like 2 plants next to each other, are homogenized by gene flow. They're not a gene pool. So I coined a new term in the chat while we were chatting of gene puddle. So we have all these little tiny gene puddles all over the place in what we call a plant species. And I just wanted to, you know, make sure that we understand the risks involved with stirring up local adaptation and mixing genotypes unless we know exactly what we're doing, which as several speakers said, we don't normally know what we're doing. So I don't know how many people heard my opening up talk this morning, but that was the point I was trying to make there, is that we can't assume, like the animal people often do, that every animal is kind of interchangeable with another one. We just have so much local adaptation and differentiation. So I throw that open to the panel.
Well, I'm really glad you brought it up because I heard your, this is Michele, by the way, Dudash. And I enjoyed your presentation this morning, and you really got me thinking about this, but I have worked closely with animal and plant people, and I think there are more similarities than differences. So I think we're going to disagree on some points, which is fine. But I also think if the unit, the taxa, whether you call it a species, a puddle, you need terminology to help land managers decide what they can do. We all don't have the luxury of this amount of detailed data. And we can, and I agree, plants are so much more interesting than animals. There's no question about it. Their mating systems, their sex, everything. So I'm totally, I transferred over from animals to plants. But I also think that plants are unique in that they're all evolving right now, and you can watch biodiversity go extinct, or you can try to manage in situations, or obviously we saw a whole breadth of presentations this afternoon where there are good units that you can monitor. So I don't think that the phylogenetic component, I mean, I think it's interesting intellectually, I'm challenged by it and I like it, but I think we have to get out of the ivory tower and think about the people with the boots on the ground and what they're doing. So I think there's room for a lot of different ideas, because I think ultimately we're all interested in trying to conserve or manage the biodiversity.
That's a really good point. We have a question about assisted migration. What about assisted migration? It is happening in California in the Sierras.
Or Michelle, is it happening? Who's assisting, may I ask? Is it, I guess I don't exactly sure what they mean.
Say on that one. It was from Vence.
Okay.
So very involved. All right, what if we move on to the next one? Assumptions that are being made in this discussion that you have to mix distant populations. Sometimes you can mix from only local sources. Morphology variations equaling heritable variation, or that differences are not due to regulatory gene plasticity. Differentiation is adaptive differentiation. Local adaptation is unique. Populations have distinct alleles, and ignores that if variation is adaptive, selection will continue to favor those alleles in the reintroduction site. So human intervention is not the end of the story. Any responses to any of those?
Well, obviously, I mean,
Yes.
I don't, please go ahead, Amy.
Oh, well, yeah. I mean, I think all of those things are true. I would say in the case study that we presented on the salt marsh, or I presented on the salt marsh bird speak, that a lot of these questions are still questions. We don't really know the answers to them. So those are, you know, hopefully next steps in the research component that goes along with managing these sites. And, you know, there is definitely for this marsh species, you know, a huge problem with sea level rise coming now. And, you know, we know in probably 100 years that all of these marshes will be more or less just barren mudflats. You know, if you look at the sea level rise projections and the GIS analyses done for our region. So, you know, understanding how to allow them to move upland if they have a place to do that. And then also seed banking and just being prepared to try a lot of heavy-handed management, I think, in restored wetland situations is going to be really necessary. So we may not have the luxury, I guess, of retaining the natural diversity as it exists now everywhere. But hopefully we can as much as possible. You know, right now, I think in our situation, there are, you know, for this species or subspecies, there are quite a few sites with multiple occurrences and relatively high genetic similarity among local sites. So, you know, there's a lot of opportunities to move locally, I think, source seeds locally.
And I'll just mention, I think all of these discussions highlight the importance of conducting reintroductions in an experimental way, at least at a minimal level, being able to track what's going in and how those populations are performing over time, so we don't lose important information to help inform future decisions, and how we tell the stories of what's going on right now to translate all of the research into more applied approaches, I think is where a lot of the work is still needed. But I really appreciate all the other speakers and all the questions so far. It's been really a great group to learn from and chat with.
We have just a couple of minutes before the break. Are there questions you would like to ask of each other? Things that you'd like to know about each other's research or things you'd like to work on?
Well, what are people's thoughts on stable hybrid zones? Some folks mentioned, introgression, but that might be one way that taxa will be able to mitigate climate change. And I was just curious about that.
So I don't know the answer for Hazardia. I don't know the answer, but I would be interested to know because of the time, that I had to devote to that study, but I would be very interested to know what the dynamics actually are between Orchidea and Squarosa there, because it seemed that potentially the species is not in the US because of introgression. I mean, but it's just a guess because they do co-occur in Baja California, but on a site-by-site basis, I don't have a feeling for that. But it was this fluke part of this study that I didn't talk about where at San Diego Botanic Garden, where some open pollinated plants, got together and we had offspring of that. So we could see that there was a certain level of hybridization, but, and then it was apparently happening in the field as well.
Mm-hmm. I mean, someone earlier made a good point that the hybrid might not necessarily serve the same role in terms of reward and so forth. So it's a really interesting question.
Yeah.
Thank you.
We have a question for Andrea. Somebody says, I might have missed this, but is there a representation of source pollination? What was the representation of source pollinations in the nursery when individual source populations weren't tracked separately? In the Washington nursery, it looks like one population was very underrepresented.
Right, yes, so that is true. Basically, when you mix before you plant individuals into a nursery setting or any setting, if you can't track individual source populations, or if you're dealing with very few maternal lines, if you can't track those maternal lines, you run the risk of having equal or the founder representation that you think is most appropriate for your restoration mix. So the less mixing that you do, or the less mixing you do until right before you do the reintroduction, the better control you can have over what goes into that reintroduction. So we definitely, it looks like one of those source populations is just not doing very well in that nursery setting. And it's, and as a result, it's not getting into the reintroductions as intended.
All right. It looks like we're kind of running slow into questions. It's a lot to think on. We do have a final panel discussion back in the main room where everybody was this morning, starting at 3:30. And Elizabeth has just added the link to the full program. So thank you all for speaking. Thank you for coming and being interested. It sounds like we have a lot more discussion to continue.
Yeah, all right, thank you everyone. And again, we'll have this recording available next week. But as Anne-Marie said, please go back to the main webinar. You'll use your original registration link that you joined this morning. And so I'm gonna go ahead and end this, but thank you everyone for being here.